Good afternoon. This video is supposed to be a revision video for tomorrow's chapter test, inshallah. And it's also a revision video for the mid-semester test, since the content of both tests is almost the same. Now, we will start by the first concept, which is the need for a circulatory system. You are frequently asked about the difference between one-celled organisms and multicellular organisms in their need for the presence of a circulatory system. So there are certain important terms that you have to use. The most important terms that you have to use is one-celled, of course, multicellular, diffusion, and transport. So you would say that one-celled organisms or unicellular organisms are surrounded by the environment from everywhere. And therefore, it's easy for components of the environment like oxygen, like nutrients, to enter through the cell membrane of such unicellular or one-celled organisms, oxygen or any kind of nutrients like glucose or salts. So you have to use the term diffusion, you have to use the cell membrane, you have to use environment, and they are in direct contact with the environment. All the parts of the organism are in direct contact with the environment. You also have to mention that just like oxygen and nutrients can enter easily by the process of diffusion, then waste substances can come out via the same process, which is diffusion. In multicellular organism, it's important again to mention the term environment. So in multicellular complex higher organisms, not all the cells are exposed to the environment. And the deeper, the more complex the organism, the more the organism has deeper cells. So I've used the term complex, I've used the term deep cells, and those deep cells are away from the environment, and therefore it's not easy for oxygen, nutrients, and all the materials needed by the organism to reach the deeper cells. And that's why there should be a transport system. That transport system should contain a fluid, for example, blood or lymph, so that the substances that are needed for the organism or the substances that the organism wants to get rid of could be easily transported in such a fluid by being dissolved. So you need to mention complex organisms. You need to mention deep cells. You need to say that the deep cells are away from the environment. The transport system carries a fluid, and the fluid is important for dissolution of those substances. The second question or type of question, questions that you might encounter on the chapter test or the mid-semester test is the components of the circulatory system. I might ask you broadly about the components of the circulatory system, and of course you would say the cardiovascular system and the lymphatic system. But if I say mention or explain the components of the circulatory system, then you would say, after having said cardiovascular and lymphatic systems, you would say that the cardiovascular system consists of cardio, of course, means the heart, vascular means the blood vessels, and then you would further explain that the blood vessels contain arteries, capillaries, and veins. On the other hand, you would explain the lymphatic system, and you would say that the lymphatic system consists of lymphatic organs and lymphatic vessels. So this is the overall structure or the overall anatomy of the circulatory system. When I say overall, I mean generalized. I don't mean, or I don't need the details. So we start by mentioning the broad categories, the cardiovascular system and the lymphatic system, and then the main subclasses of each the heart, the blood vessels, arteries, capillaries, and veins, and the lymphatic system, lymphatic organs, and lymphatic vessels. I might ask you afterward about the general function of the circulatory system or the overall function. So um, just as I asked about the overall anatomy, I can ask about the overall function of each of them. Of course, for the cardiovascular system, the main function, as we said before, is can be analyzed from the organs of the cardiovascular system. So you would talk, for example, about the pumping action, which is the action of the heart. So the heart pumps blood under pressure, and this blood under pressure can reach the tissues and therefore can do the second function, which is delivery of oxygen and nutrients to the tissues. And at the same time, the presence of capillaries allows the exchange, whether the exchange at the level of the tissues, which is the tissue capillary exchange, or the level of the alveoli, which is the alveolo-capillary exchange. And both are important. The exchange at the tissues, the tissue capillary exchange, allows the tissues to get rid of their carbon dioxide and waste and to intake oxygen and nutrients. At the level of the alveoli or the alveolo-capillary exchange, it allows unloading of the blood from carbon dioxide and loading of the blood with oxygen. On the other hand, the overall, so that's the main function. It's not enough, of course, to say that the main function of the cardiovascular system is transport. It's true, it's correct, but transport requires first a pump to generate pressure to push the blood forward, and requires at the end of the day a process of exchange in order to unload and load. On the other hand, the overall function of the lymphatic system is essentially that of the lymphatic organs and that of the lymphatic vessels. For the lymphatic organs, of course, the main function is number one, a production of lymphocytes, if it is a primary lymphatic organ. Second, it might be concerned with filtration of blood from pathogens. And the function of the lymphatic vessels is to transport the surplus fluid. So it's important for homeostasis. So you can write all the functions of the lymphatic system in a very brief way. So when do we write the functions briefly? We write, or generally, when we are asked about the overall function. When do we say the anatomy briefly? When we are asked about the overall anatomy. This is important to answer exactly according to what's required in the question, because if you don't do this, if you write full details, you might lose time and you might not be able to finish your test. Of course, I can ask you to enumerate the lymphatic organs. So the lymphatic organs include, you can start from above downward in order not to miss anything. You can start first by the lymph nodes, because the lymph nodes are present everywhere. You know that we have groups of lymph nodes, and therefore they are present everywhere. And so you start by the lymph nodes, and then you mention in the oropharynx and in the nasopharynx, you have the tonsils and the adenoids. Here behind the sternum, you have the thymus. Here you have the spleen in the left upper quadrant of the abdomen. And then in the intestines, in the mucosa of the intestines, you have Peyer's patches. You have the liver as a primary lymphatic organ in the right upper quadrant of the abdomen, and you have the appendix, and of course all over the body inside the long bones you have the bone marrow. So you answer briefly when you are asked to just enumerate or list the lymphatic organs and mention their names. I might also ask you about the hierarchy of the lymphatic vessels. So if I ask you about the hierarchy of the lymphatic vessels, then you can start by the smallest and say, for example, that we have the lymphatic capillaries, So you can say, for example, that you start by the lymphatic capillaries, and then you have larger lymphatics, and then you have the two main lymphatic ducts, where all the large lymphatics pour their content of lymph, the thoracic duct and the right lymphatic duct. So the hierarchy of the lymphatic vessels would start by the lymphatic capillaries, and then the small lymphatic vessels, and then the medium-sized lymphatic vessels, and then the large lymphatic vessels, and then the two main ducts, which are the thoracic ducts and the thoracic duct and the right lymphatic duct. I might also ask you about the fate of the lymph inside the thoracic duct and the right lymphatic duct. So both of them will pour into the subclavian veins. The right lymphatic duct into the right subclavian vein, and on the other side, the thoracic duct into the left subclavian vein. Now, what's the main difference in structure between, another question, what's the main difference in structure between the lymphatic vessels and the blood vessels? First of all, the lymphatic vessels are very thin-walled, meaning that they do not have a muscular wall like in arteries and veins, for example, even the large ones. And they contain valves all along their course. And we know that this is very different from the blood vessels, because we know that none of the blood vessels, except the veins, of course, has valves along its course. So only veins have valves along their course, because we will explain why. So they are valve containing along their course, and they are very thin walled. And in addition to this, they, as we said, they have valves all along their course, and they are thin-walled. And in addition to this, they have fenestrations, and those fenestrations are wider than the fenestrations in the blood vessels, and those fenestrations in between their endothelium allow them to intake the surplus fluid that cannot be taken by the veins. So why do we call them secondary circulatory system? Yani why do we say that the lymphatic vessels are considered a secondary circulatory system? This is because all the lymphatic vessels run parallel to the veins. So they go hand in hand with the veins, and they carry the surplus fluid that cannot be carried by the veins, and that will form later on the lymphatic fluid. What's the difference between the lymphatic, a third question here on the slide, concerning that paragraph? What's the difference between the lymphatic vessels and the blood vessels? As we said before, the lymphatic system does not have a pump, whereas the heart has a central pump. And because it does not have a pump, so if you have fluid that is going upward against gravity, then it's very liable to pull down if there weren't protective mechanisms, because there is no central pump. How is the central pump important? The central pump generates pressure, and that pressure is capable of pushing the fluid upward against gravity. So if you don't have a pump, it would be very difficult for the lymphatic fluid to go upward against gravity. That's why there are three other mechanisms that, or actually two other mechanisms that help this forward flow, helped by the valves. The first one is the presence of muscle contraction, as you see on the slide, muscle movement around the lymphatic vessels. So if you have the muscles here and there, all around the lymphatic, so the contraction of the muscle squeezes the lymphatic vessel, milks it up, and helps the lymphatic fluid to move upward against gravity. The second thing is the presence of the arteries besides the skeletal muscles or inside the skeletal muscles. We have arteries, but these are small arteries. But I'm talking about the large arteries that run hand in hand, or the larger actually arteries that run hand in hand with the lymphatic. So the pulsations of the arteries also help squeezing up the lymph and help causing contraction of the lymphatic vessel. It is as if the lymphatic vessel was contracting, but actually the inward movement and the outward movement is due to the pulsations of the artery from one side and to the contraction of the muscle from another side. These two factors, the factor related to muscle movement and that related to the pulsation of the artery, are helped by the valves. What is the importance of valves that are present along the lymphatic vessels? These valves ensure one-way flow of lymph upward against gravity, or when I say forward, I mean in the wanted direction, in the needed direction. So they ensure the forward movement of lymph towards the destination of lymph and prevent backflow. In the circulatory system, we have a pump, so the arteries do not require valves. Blood capillaries do not have valves. Valves are only present in the veins for the same reason, to ensure forward flow, this time not of lymph, but to ensure forward flow of blood and prevent backflow. At the beginning of this slide, we talked about the hierarchy of lymphatic vessels, but there is another question, which is the types of lymphatic vessels. We can, of course, classify the lymphatic vessels in different ways. One of the ways is to classify them according to their function. So we have the afferent vessels, the efferent vessels, and the lacteals. So when are you required to mention afferent, efferent, and lacteals when you're asked about the types of lymphatic vessels according to their functions? So the afferent vessels will carry lymph towards the lymph nodes. The efferent vessels will carry away from the lymph nodes, and the lacteals are the ones that are responsible for absorption of dietary fat from the contents of the small intestine. How are lacteals different? How are lacteals well adapted to absorb fat? Lacteals have larger fenestrations. When they have larger fenestrations, they can absorb larger molecules, for example, medium chain fatty acids, or even larger molecules. Here we can see a picture of the lymphatic ducts. We have the right lymphatic duct, as we said, that empties in the right subclavian vein, or actually here at the junction of the internal jugular vein and the subclavian vein. This is the subclavian vein, and that is the vertical one is the internal jugular vein, and the horizontal one is the subclavian vein. On the other hand, the thoracic duct empties at the junction of the internal jugular vein and the subclavian vein, but on the left side. We didn't mention the cisterna chyli, which is like a widened part of the thoracic duct in the most inferior part of the thoracic duct. You needn't know it, by the way. Now, if I ask you not about the overall function of the lymphatic vessels, but about specific functions of the lymphatic vessels, you would mention these three functions. Homeostasis through maintaining fluid balance. So, maintaining fluid balance, of course, is a part or an aspect of homeostasis. We have just mentioned absorbing fats from the digestive system, from the small intestine, and number three, the immune function. As for maintaining fluid balance, we know that there is in between the cells what we call the intercellular fluid or the interstitial fluid. This fluid is present in between the cells. And this fluid is due to passage of fluid outside the capillaries through the fenestrations at the arterial side of the capillaries. But at the venous side of the capillary, so if we have, this is the capillary bed, so this is the arteriole and this is the venule. So here the capillaries still have pressure, high pressure. So there is fluid that exudates outside. And here at the venous side, there is fluid that re-enters to return to the heart. So if the fluid that is exudated outward is more than the fluid that is reabsorbed inward, then we will have a surplus fluid, and that surplus fluid is usually carried by the veins. But if the veins cannot carry all this surplus fluid, then the lymphatics should collect this excess fluid, because it's not only water, but it's water with waste substances and debris from the tissues. So what happens? The second piece of information here tells you what happens if the lymphatics, the regional lymphatics in a certain place, do not absorb all the fluid that should be absorbed. We will get what we call lymphedema. Edema in general means swelling, and lymphedema is a particular type of edema due to dysfunction of the lymphatic vessels, and it is characterized by being non-pitting, as we said before in class. The second function is the function of the lacteals. You know that absorption occurs in the terminal part of the small intestine, which is the ileum, and that the soluble digested substance pass to the blood capillaries around the small intestine. But in order for them to be able to be absorbed into blood, they should be what? They should be water soluble. If they are not water soluble, then they cannot dissolve in water, then they cannot dissolve and be transported in plasma. This is the case of fats. Fats are not water soluble, and therefore they cannot be absorbed through the blood capillaries, and they are absorbed via the lacteals. So the fats in the small intestine, especially the medium-sized molecules and the large molecules, have a special a way of transport or absorption, and it is through the lymphatics. And of course, all this content of the lacteals and all other lymphatics will join at the end of the day the systemic general circulation, and they will reach the blood. The third function of the lymphatic vessels is an immune function. I might ask you about each of these items separately. How do lymphatic vessels maintain fluid balance? How do lymphatic vessels help the process of absorption? How do lymphatic vessels help immunity? They help immunity, of course, because the lymph is full of lymphocytes. It's not only water, it's not only debris, it's not only salts, it's also full of lymphocytes. And those lymphocytes circulate in the whole body. They detect any pathogens, and this is what we call immune surveillance. Immune surveillance means that they are alert in case a pathogen or a foreign body enters. They start multiplying and they start secreting substances to fight this infectious organism. One of the major components, as you know, other than the lymphatic vessels, we are now talking about another major component, which is the lymph node. And we said before that the lymph nodes are small. They are less than 1 centimeter usually, since they are normal. They are scattered all over the body in the form of groups. And those groups are called regional lymph nodes. So every particular region in the human body has a group of lymph nodes, as we said before. So, for example, if we look at the diagram here, we can see all these lymph nodes that lie in the neck are called the cervical lymph nodes. And the cervical lymph nodes, of course, they drain from the head and from the neck. Cervical means related to the neck. And as you can see, they are sub-classified into groups, an anterior group, a posterior group, a group at the bottom, a submandibular group, and so on. You needn't know the labeling on or the labels on that diagram, but you just have to know that those lymph nodes that are present in the region of the neck are called cervical lymph nodes, and that they drain lymph from the head and the neck. Sorry. We also have very important lymph nodes, which are the axillary lymph nodes or the armpit lymph nodes. Of course, armpit is an informal term. The axillary lymph nodes are very important because they drain from the chest wall, they drain from the breast, and they drain from the lungs and the inside of the chest cavity. So if they drain from the breast, then regular, that's why there is a justified question, regular checkup every 6 months on those lymph nodes is important because this helps early. If they are enlarged, you would start investigating, and this would help you early diagnosis of cancer breast, for example, and the early diagnosis of cancer breast can save the life of the patient. But we also have other groups like, for example, the abdominal lymph nodes. I'm not sure whether we have a picture of them or not. Yes. So we have here the axillary lymph nodes again, and then you need not know, of course, the details of their particular locations. And we have here, as you can see, the para-aortic lymph nodes, which are the abdominal lymph nodes, and you have the pelvic, lower down in the pelvis, we have the pelvic lymph nodes. Of course, the para-aortic lymph nodes will drain from the abdomen, and the pelvic lymph nodes will drain from the pelvis. That's why there is regionality in the distribution of the lymph nodes. The lymph nodes are important because of many reasons. Why are they important? Number one, they are full of lymphocytes, and therefore they are responsible for immunity, and specifically the adaptive immunity, because we know that most lymphocytes are responsible for specific adaptive immunity. Only a few of them, like for example the T helper cell, can help both the B lymphocytes and the T lymphocytes, and can warn or alert the non-specific cells like the macrophages, for example, to start their job. So the T helper cells can work both ways, in the innate immunity and in the adaptive immunity. And we'll be saying now the difference briefly between the two. So first of all, they are responsible for mainly adaptive specific immunity because they are full of B lymphocytes and T lymphocytes. And second, they help filtration. And here filtration is just a mechanical filtration first, because the afferent lymphatics pour their content of lymph into the lymph nodes. So they are important to filter the lymph. And when they filter the lymph, this means that they trap the pathogens, they detect the pathogens, they attack the pathogens. So again, this is an immune function. So they contain white blood cells, B and T lymphocytes, as you can see on the slide, and they act as filters for the lymphatic system. And here, filter does not only mean trapping the microbe, but it's trapping and attacking, because the trapping has been done by a cell like the B lymphocyte or the T lymphocyte. What kind of foreign particles or invaders can be attacked by the lymph nodes? Plenty of things. For example, pathogens like viruses and bacteria, or on the other hand, malignant cells like cancer. So they filter and attack, they catch and attack viruses, bacteria, and other pathogens, but they also can detect and attack cancer. So if your immune system is not working well, this means that cancer might spread easily in your body. What's the importance of trapping the microbes or the cancer cells, preventing them from reaching the bloodstream? So it's okay if the virus or the bacterium or the cancer cell is still in the lymph node, as long as it did not reach the bloodstream. So the main purpose of the lymph nodes is to prevent the pathogen or the cancer cells as much as they can from reaching the bloodstream. Now, of course, the definition of lymphadenopathy is very important, and it's important to know the difference between lymphadenopathy and lymphoma. Now we are focusing on lymphadenopathy, which is more inclusive, which is a more general term, lymphadenopathy. Because lymphadenopathy means a swollen lymph node in general, whether the swelling of the lymph node, we have, of course, several factors. Number one, when they drain an infection or an inflammation. So inflammation is a factor. Number two, autoimmunity, of course. And number three, if we have cancer in the body. So these are three major causes of lymphadenopathy. So they might be swollen because of an infection. They are fighting an infection. They are fighting a tumor, a nearby tumor, or there is a nearby injury. If there is a nearby injury, how is this related? Because inflammation might be injurious or might be due to an injury or a trauma. So it might be traumatic, but inflammation might be due to an infection. So when I say that inflammation results in lymphadenopathy, I mean inflammation of a nearby tissue from tissue injury or inflammation due to invasion of the body by a pathogen, as you know. But autoimmunity might also cause lymphadenopathy, because you know that in autoimmunity, the immune system is attacking self cells. So when the immune system is attacking self cells, then the response of the immune system is not normal. The response of the immune system is exaggerated. And that's why the lymph nodes keep on proliferating and dividing in order to cope with this exaggerated reaction of the immune system. In cancer, there are two causes for enlargement of the lymph nodes, actually. The first cause is that cancer has reached the lymph node. And in such a case, of course, there are malignant lymphocytes inside the lymph node, and the malignant lymphocytes rapidly divide. They divide very, very rapidly, and therefore there is swelling. The lymph node enlarges because it has been reached by malignant cells. But the other possibility is that the body has discovered a malignant cell elsewhere, and that there is activation of the immune system to fight cancer even before cancer reaches the lymph nodes, even when cancer is still in the tissues or still in the blood. Actually, the lymph nodes might swell in order to allow the immune system to fight. But more commonly, in cases of cancer, the enlargement is due to rapid cell division and rapid proliferation of the abnormal cancer cells. Now, how do the lymph nodes look like in lymphadenopathy? Of course, first of all, by definition, there is swelling. So, number one, there is swelling. Number two, there might be pain. And pain, by the way, is reassuring, because usually when we have infection, the patient suffers from pain or complains of pain. Pain is a symptom, as you know. Tenderness is what the medical doctor medical doctor like discovers on examination. You are examining the lymph nodes and you feel the patient shouts out of pain, or just the facial expressions of the patient show that he is in pain. So the common causes of swelling first are infections, viral, bacterial, like cold, like flu, even ear infections. Of course, if you have ear infections, this is drained by the cervical lymph nodes. If you have a skin infection, the regional lymph node that drains will depend on the site of infection on the skin. But there are other signs for lymphadenopathy. For example, that the consistency. Here we talked about the size, they are swollen. We talked about the presence or absence of pain, and then the consistency. Usually the lymph nodes are firm and rubbery, but in case of cancer, they are hard. The fourth option is the skin above the lymph node. Usually the skin above the lymph node is free if it is not a malignant lymphadenopathy or lymphoma, but it might be fixed to the lymph node. It might be fixed to the lymph node in case there is malignancy. So when is lymphadenopathy serious? If you have the following signs. Number one, if it is hard. Number two, if it is fixed to the overlying skin. Number three, if it is painless. Number four, if it persists for more than 2 to 4 weeks. Number five, if it is associated with systemic symptoms like night sweat, night fever, or unexplained weight loss. Of course, as for the histology, the cellular structure, histology means cellular structure. You can talk about the anatomy of an organ or a body system, but when you talk about a tissue, you say histology of a tissue. And when I say histology of a tissue, I mean the types of cells and the structure of those cells. So here you can see that we can see the lymphocytes arranged in lymphatic follicles, spherical follicles, and in between them we have trabeculae, or like separations made of connective tissue in between the lymph nodes. I'll not ask you about the histology of the lymph nodes. I'll not ask you to label a diagram or to draw a diagram about the lymph nodes. And now we come to the lymphoma. Of course, the lymphoma is a malignant disease, and it is considered not only a malignant disease affecting the lymph nodes and the lymphatic vessels, but it is also considered blood cancer. Why? You have to justify this, because the malignant lymphocytes originate essentially in the bone marrow and in which is the site of formation of all the blood components. And those malignant lymphocytes will circulate in the lymphatic vessels and will circulate in blood, because the bone marrow is richly supplied with blood. So actually it's a cancer of lymph nodes, and blood. Since it is malignant, then it will have all the symptomatology and the signs of malignant lymphadenopathy that we have just mentioned on the previous slide, meaning that it starts painless, and then later on it becomes painful. The skin might be fixed to the lymph nodes. It might be pitted. Sorry. It is associated with systemic symptoms like fever, night sweat, unexplained weight loss, and sometimes there is itchy skin, chest pain, and shortness of breath. The chest pain and shortness of breath are mostly due to compression by the lymph nodes that are the enlarged lymph nodes, compression by enlarged lymph nodes. If they compress the airways, then they will cause shortness of breath and chest pain. But itchy skin is due to certain chemicals similar to histamine that cause an allergic reaction on the skin. Actually, as we said before, we have two types of lymphomas depending on the presence of this cell, very specific cell, which is called the Reed-Sternberg cell. And if the Reed-Sternberg cell is present, then it's Hodgkin's lymphoma. If not, then it is non-Hodgkin's lymphoma. So this is the first difference between Hodgkin and non-Hodgkin's lymphoma. The second difference is the incidence, the frequency of occurrence. Of course, non-Hodgkin's lymphoma is more common, is more prevalent, and it consists of about 70 subtypes. And that's why scientists divided or categorized non-Hodgkin's lymphoma into low grade and high grade. The low grade, by low grade, we mean low risk. Low grade here means low risk because it is slowly growing, so it does not spread rapidly all over the body. And we have a high risk, which is the other group that divide rapidly and spread to the whole body. And as you know, lymphomas are divided into stages according to where they are present. So if they are local in the regionally, then this is stage one. If they spread so if they stay in the, for example, if you have cancer breast, for example, so if you are still in the organ, so this is stage one. And then if it spreads to the regional lymph nodes, then it is stage two. And then it depends on whether it is on one side of the diaphragm or on both sides of the diaphragm, and then spreading all over the body. This is stage four in different organs of the body. But the stages differ because there are some classifications that start the classification with zero. I'll not ask you about the stages of cancer, but it's good to know that it's classified according to whether it is local in the organ or if it's spread to a regional lymph node, or if and then when it spreads, are the lymph nodes on one side of the diaphragm or on both sides of the diaphragm, and then whether they are spread all over the body in different organs of the body. The exact cause, the etiology, I might ask you about the underlying cause or the predisposing factor. So I might ask you about the cause, or I might ask you about the predisposing factors. So the predisposing factor is mainly genetic. It's mainly a gene mutation. Some people are naturally predisposed to getting the lymphoma because they have this genetic mutation that causes them to, or that results in the conversion of the lymphocytes from benign, normal, well-functioning lymphocytes to abnormal lymphocytes. But there are other acquired factors, like, for example, certain viruses, like the Epstein-Barr virus, the human immunodeficiency virus, and also autoimmune diseases may be followed later on by the occurrence of lymphoma. I just want to clarify something that when I say that lymphoma is due to a gene mutation that results in a DNA abnormality. So, for example, if this is the mother cell that forms the lymphocyte and this is the nucleus, and here we have the DNA. So, if there is a change in the DNA, then this is a change in the gene inside the nucleus of the lymphocyte. And this DNA mutation will result in the formation of an abnormal lymphocyte that will be malignant at the end of the day. But what I want to clarify is that this mutation can occur during the life of the individual. For example, if the individual has been exposed, for example, to X-rays or to ultraviolet rays. But in order for this mutation to occur, you should be genetically predisposed, meaning that you already have in your genes that you inherited from your parents some genes that are not fully normal and that give you this predisposition to the occurrence of lymphoma. Another thing that I want to clarify, although I'll not ask you about the stages of lymphoma, that there is an easy way to remember the stages if you use the stage that starts by 1. So not the stage that starts by 0. 0 is usually when the lymph node, as I said on the previous slide, are confined to the organ. So if you use 1, 2, 3, 4, an easy way to remember is that stage 1 is when it is confined to the regional lymph nodes only, meaning that you have only one group of lymph nodes. In stage 2, you have two groups of lymph nodes, but they are all on one side of the diaphragm, either above the diaphragm or below the diaphragm. In stage 3, you have both ways. You have above and below the diaphragm, so it's more serious. And at the end, you have stage 4 that is widespread in the body. So yeah, I thought this could be an easy way to remember the stages of lymphoma. As we said before, lymphoma is considered a systemic disease because this is a justify question here, justify because the malignant lymphocytes circulate both in the blood and in the lymphatic vessels. We also said that the origin is that the lymphocytes, which are the B cells or the T cells or even the non-specific natural killer cell, undergoes mutation. And as I said on the previous slide, that mutation occurs during the life of an individual, but this individual should be genetically predisposed. Of course, when you have what's the impact of having malignant lymphocytes inside the lymphatic vessels? Of course, these malignant cells will accumulate in the lymphatic organs. So the main problem with lymphoma is that you have malignant lymphocytes, or you start by having mutant lymphocytes, but those mutant lymphocytes, as usual, and as all normal lymphocytes do, they will aggregate in the lymphatic organs. And this is the seriousness of lymphoma. So if I ever ask you why lymphoma is a serious disease, lymphoma is a serious disease usually because number 1, it is systemic, and number 2, because the circulating lymphocytes will settle in the lymphatic organs. And that what makes it, this is an additional cause why it is systemic. It is systemic because the lymphocytes circulate in blood and lymph vessels, but also systemic because it is widespread in the body, since all the lymphatic organs will carry malignant lymphocytes. And the seriousness is that these organs that are packed with malignant lymphocytes, of course, will never do their normal function. The last thing we would say about the information on that slide is the relationship between lymphomas and leukemias. Leukemia is also a type of cancer, and it is also a type of blood cancer that originates in the white blood cells, but in another type of white blood cells, and it does not start in the bone marrow. It starts in the it does not start in the lymphatic system, but it starts in the bone marrow. So in leukemias, we have a malignancy. It's again a blood malignancy, but blood only. It's blood malignancy, okay? And the abnormal cells originate in the bone marrow. And moreover, we can say that the cells are the polymorphonuclear leukocytes, PNLs, and not the lymphocytes. But at the end of the day, you will have, because there is a connection between the blood and between the lymphatic system, you might find out that the lymph nodes in case of leukemia are also enlarged. But the origin in case of leukemia is the bone marrow, not the lymphatic system. Why is lymphoma harmful, as we said before, and why is it associated with enlargement of the lymph node? Enlargement, of course, because the malignant lymphocytes undergo rapid, uncontrollable mitosis or rapid, uncontrollable cell division and results in huge swelling of the lymph nodes, and they might reach a very big size and they might fuse together because of the large size. How can we treat a case of lymphoma? You can use chemotherapy, you can use radiation, you can use immunotherapy, and you can also use stem cell transplants. We haven't explained stem cell transplants in detail. That's why I will not ask you about them in the test or chapter test or mid-semester test, but we mentioned chemotherapy and we said that there are two ways chemotherapy can affect the lymphocytes. The first one is by causing DNA damage to the cells, and the second one is by preventing spindle fiber formation. And we said that the spindle fibers are important for arrangement of the chromosomes at the metaphase plate during metaphase, and therefore if the chromosomes are not fixed in place, anaphase will not occur, separation into two daughter cells will not occur. Radiation, of course, the energy of radiation would destroy the DNA of the cell again, and immunotherapy here we use specific antibodies, and these specific antibodies specifically attack the antigens that are present on the malignant lymphocyte. So if this is the malignant lymphocyte with its abnormal antigen on the cell membrane, so the antibody, the specific antibody, will kill only the cell that carries the antigen. And the importance of immunotherapy, which is, by the way, a very expensive type of therapy, is that we attack only the cancer cells specifically. There is a famous question, of course, about the difference between a primary cancer and a secondary cancer, and don't confuse it with a primary lymphatic organ and a secondary lymphatic organ. As we said before, a primary lymphatic organ is an organ where the lymphocytes are produced and get matured, whereas a secondary lymphatic organ is an organ where the lymphocytes are stored and are allowed to proliferate. But a secondary organ, lymphatic organ, can never form lymph nodes from scratch and can never allow them to mature. It's only a site of storage and a site that allows reproduction or mitotic cell division. This is concerning primary organ and secondary organ. But we also have another couple, which is primary cancer and secondary cancer. The primary cancer is the cancer that first originated in an organ. The secondary cancer is the metastasis, when some malignant cells break off from the original tumor and they circulate in lymph or sometimes in blood to reach other areas of the body. We also talked about the spleen as a lymphatic organ, but it's worth mentioning the differences between the spleen and the lymph nodes. Both the spleen and the lymph nodes contain lymphatic follicles. They are spherical collections of lymphocytes like this. So we have the lymphocytes in a spherical shape. So this is a lymphatic follicle. The lymph node is made only of those lymphatic follicles, and in between we have connective tissue trabeculae or septa. But in the spleen, you have those lymphatic follicles in the white pulp of the spleen, but you also have the red pulp of the spleen that is made of dilated blood vessel segments that are called the blood sinuses. So a major difference between the spleen and the lymph node, although they are both lymphatic organs, is that the spleen contains blood. Blood passes inside the spleen, and lymph also passes inside the spleen, but lymph passes through the white pulp and blood passes through the red pulp. So the common thing is that both are lymphatic organs, both have lymphocytes, both trap pathogens and attack them and provide immune surveillance for the body. But in addition to this, because of the red pulp in the spleen can store platelets, the spleen can store blood vessels, red blood corpuscles, and the spleen can also destroy old red blood cells or damaged red blood cells. Where is the spleen located? We said it is in the upper left quadrant. And of course, since the spleen contains, if I ever ask you about the immune importance of the spleen, since the spleen contains the white pulp, then it contains lymphocytes, then it is responsible for immune actions of the body. When I ask about the storage functions of the spleen, you will say that the spleen is important for storage of blood, red blood corpuscles, for storage of platelets. So what's the effect of splenectomy on a patient that has an enlarged spleen because of a lymphoma, for example, or because of any other reason? Of course, if the splenectomy means surgical removal of the spleen, but if the patient has removed the spleen surgically, then of course immunity will be affected because a good deal of lymphocytes will be absent from the body, and therefore they wouldn't carry out their original immune function. So they are at a greater risk of getting infections. Of course, there are other problems related to the red pulp and the ability of the spleen to sequester and isolate the blood platelets and store them. So there are problems related to the platelet count, but we haven't discussed them in class. I would just like to clarify something or highlight something concerning the difference between lymphoma and leukemia. As we said before, both are systemic cancers, and both might be associated with enlarged lymph nodes, and both are associated with mutations in the DNA of the lymphocytes. But most of the lymphomas originate, although the lymphocytes themselves, this is the confusion, although the lymphocytes develop in the bone marrow, because the bone marrow is a primary organ, so the lymphocytes develop in the bone marrow. The bone marrow is the main site for hematopoiesis. So because the bone marrow is the main site for development of the blood cells in general, so the lymphocytes develop in the bone marrow. But in most lymphomas, the abnormal lymphocytes develop after they leave the bone marrow. The malignant lymphocytes develop in the secondary lymph organs, like the lymph nodes, for example, and the spleen. But in leukemia, although the white blood cells of leukemia also originate in the bone marrow, yet the abnormality or the mutation that occurs to them starts in the bone marrow itself. So a major difference between lymphoma and leukemia is where the malignancy started. In most lymphomas, the malignancy does not start in the bone marrow itself, although the bone marrow is the organ that produced the original lymphocytes, but the mutation occurs in the secondary lymph organs after the lymphocytes leave the bone marrow, whereas in leukemias, the malignant cells and their mutation occurs inside the bone marrow. The liver is the main lymph producer, as we said before, but it's not because it is a primary lymphatic organ. It's not. It is a secondary lymphatic organ. It is because it filters a lot of fluid, and therefore it is responsible for the fluid component of the lymph. The liver contains several important cells that, like the natural killer cells, the natural killer T cells, and the antigen presenting cells. As we said before, there is a difference between innate immunity and adaptive immunity. Innate immunity. So there is a difference, I'm sorry, there is a difference between innate immunity and adaptive immunity. Innate immunity is a non-specific type of immunity that is directed towards any foreign antigen, whereas adaptive immunity is a specific immunity that develops in response to a particular antigen or to a specific antigen. Of course, the most famous cells for adaptive immunity are the B lymphocytes and the T lymphocytes. For the innate non-specific immunity that represents the first line of defense in the body, we have the natural killer cells, we have the natural killer T cells, and we have the antigen presenting cells. The main difference between natural killer and natural killer T is that the natural killer does not have any receptors on its surface, but the natural killer T does not have any receptors similar to those of the T cells. I mean, of course, there are other receptors, but in the natural killer T cells, there are certain receptors that are very similar to those of the T cell. We call them T cell receptors, and therefore the natural killer cell is considered like a bridge between non-specific immunity, innate immunity on one side, and specific immunity represented by the T cells on the other side. The antigen presenting cells are mostly macrophages. They are large cells that have like feet-like processes of their cytoplasm. They engulf the microbe, they engulf the foreign body or the antigen, and then after engulfing it, they digest it into small fragments, and then they have receptors on their surface to present that antigen to the T cells and to the B cells, so that the B cells and the T cells get warned that there is a foreign antigen in the body, and they start dividing to increase their number. And therefore, the antigen presenting cells are very important to stimulate the immune system and warn it beforehand that there is a pathogen or an intruder inside the body. So they are very important for immune surveillance, those antigen presenting cells. And some of those antigen presenting cells are responsible for controlling the immune system and causing what we call immune tolerance, teaching the immune system or teaching certain immune cells to be tolerant. What do we mean by tolerance? We mean that the immune cells are taught not to attack self. So self is not allowed to be attacked. And there is a very famous cell, maybe we didn't mention it, that is responsible for tolerance. For example, tolerance to ingested antigens, as I said before, bananas, strawberries, chocolate. You're not supposed to consider them intruders or foreign antigens. So there is a cell called T regulatory cell, and the T regulatory cell is a specific type of T lymphocytes whose main function is to regulate the immune cells, kill all the cells that are not immune tolerant, that want to attack self and favor the cells that are immune tolerant, and that understand that they are not supposed to attack self cells. As we said before, the bone marrow is a primary lymphatic organ, but it's not only. When I say primary lymphatic organ, I mean the bone marrow produces lymphocytes, and that it allows maturation or maturity of some of these lymphocytes, especially the B lymphocytes. But the bone marrow has another function, which is acting as a hematopoietic organ, because hematopoiesis means synthesis of the blood cells, hematopoiesis. And what allows the bone marrow to carry out hematopoiesis is that the bone marrow contains undifferentiated stem cells. So this is what allows the bone marrow to carry out hematopoiesis, and also this is what allows the bone marrow to produce lymphocytes and be a primary lymphatic organ. But the third function of the bone marrow is that it allows B cell maturation. And in order for the cells to mature, there should be a very comfortable environment, a very suitable environment, which we call the critical environment. You need this critical environment for making the immune cells and for teaching the immune cells. So as we said before, the bone marrow is a primary lymphatic organ, but the bone marrow and the thymus both are considered primary lymphatic organs. And of course, when I say that the bone marrow is a primary lymphatic organ, or that the bone marrow carries out hematopoiesis, I mean the red bone marrow before the deposition of fat. So both B lymphocytes and T lymphocytes are formed in the bone marrow, but B lymphocyte maturation occurs in the bone marrow, whereas T lymphocyte maturation occurs in the thymus. But the bone marrow also acts as a reservoir for immune cells, because after the first attack, there is development of what we call the memory T cells. The memory T cells stay in the bone marrow and in the circulation, and their main function is immune surveillance. So upon the detection of any secondary exposure to the antigen, they wake up the immune system, they trigger the immune system to be activated more rapidly, to produce more antibodies, to produce more cytokines. So the secondary exposure to the antigen, the response of the immune system is usually very rapid and exaggerated, and that's why you are now said to be immune to that virus or immune to that bacterium. You don't get the disease once again because of the memory T cells. So the importance of the memory T cells is that they remember the pathogen, and once they see it upon secondary exposure, they elicit a very rapid immune response and therefore prevent the occurrence of the disease. So we say that you are immune to the disease, meaning that you cannot catch it once again. And then we also have, similarly to the memory T cells, we have long-lived plasma cells. The plasma cells are cells that have nuclei like this, and the plasma cells produce antibodies. Long-lived plasma cells also are plasma cells that stay in the circulation for a long period of time, and those plasma cells remember the microbe and produce antibodies, a big amount of antibodies upon secondary exposure. We talked, of course, about hematopoiesis because of the presence of the stem cells, and hematopoiesis means production of all types of cells, red blood cells, white blood cells, and platelets. And the bone marrow actually is the spongy tissue inside the bone, as I said before. If this is the bone, the outside is hard, but the inside is highly vascular and spongy. Spongy means that it contains spaces to accommodate the blood cells. Of course, the red bone marrow is the one that carries out hematopoiesis. The yellow bone marrow is important for energy storage. Um, I might ask you that question that is a bit difficult. What's the interaction of the bone marrow with the other lymphatic organs? All the blood cells that are produced in the bone marrow are released from the bone marrow, and they go either to the blood or to the lymph. And therefore, all the lymphocytes that populate the secondary lymphatic organs arise primarily from the bone marrow. Is the bone marrow important for immune response? Of course, since the bone marrow produces B lymphocytes and produces T lymphocytes and produces even the cells of the innate immunity like natural killer cells and antigen presenting cells. And by the way, you should know the difference between MHC1, MHC2, and CD1. MHC1 is a molecule that is present on the cell membranes of all your cells, and it is like your ID, and it differs from one person to another. And as we said before, it is important for tissue matching. If you want to donate, for example, an organ like the kidney, you cannot donate unless there is matching between your MHC1 and the MHC1 of the recipient. We can also call MHC1 molecules. We can call them in human beings only HLA, human leukocytic antigen, because they belong to the human species. They are present on the leukocytes, but they are also present on other cells, and they are the most important molecules that differentiate self cells from non-self cells. On the other hand, MHC2 and CD1 are like molecules or receptors they are protein molecules that are present on the surface of the antigen presenting cells, and that allow them to present the processed parts of the processed antigen. So we call them receptors because they receive the processed antigen actually. So the MHC2 receives processed antigens that are peptides, as we said before, but the CD1 receives antigens that are, so if you have a CD1 here, CD1 receptor or molecule, it will receive processed antigens if they contain lipids or fats, as we said in class before. The thymus is considered a primary lymphoid organ. It is behind the sternum, and we said that it is important. It's true that it does not synthesize the lymphocytes, but it is a site for maturation and development of the lymphocytes and selection of the T lymphocytes. This is very important. So it's not just a place where the lymphocytes are collected and stored, but it's a functional place where the lymphocytes are taught to be selective, meaning that they are taught to be self-tolerant. So what is self-tolerant? Self-tolerant, as we said before, means that these lymphocytes are taught not to attack your own body cells. It's made up of an outer cortex and an inner medulla. The outer cortex contains the immature cells, and the inner medulla contains the mature cells. And it does not have afferent lymphatic vessels. Maybe we didn't say this in class. As we said before, there is positive selection and negative selection. Positive selection means that the cells that are favorable, the cells that are self-tolerant, will reproduce, and the cells that are reactive, self-reactive, will be exported to the periphery. So T cell education means that within the thymus, T cells undergo positive and negative selection. What's the importance of positive and negative selection? It ensures that only the lymphocytes that are tolerant, only the lymphocytes that are non-self-reactive, because they are not supposed to, will stay. And I'm sorry, I made a mistake. The cells that are non-self-tolerant will be exported to the outside of the thymus. Of course, we cannot export a T lymphocyte that is not self-tolerant. So this is positive selection. You are self-tolerant, you will not destroy your cells, then you will be allowed to be exported to the periphery of the thymus and then to the body because you are functioning correctly. The other ones that are not self-tolerant will undergo negative selection and will be destroyed. Is the thymus an endocrine gland? Partly yes, because it contains or it secretes a hormone, which is thymosin hormone, that promotes the maturation of the T lymphocytes, of course, only because it's only the T lymphocytes that mature in the thymus. But later on, after childhood, the thymus undergoes involution. What is involution? It's diminution in size after puberty, and it is replaced by fat as we grow older. Sometimes the dysfunction of the thymus results in either immunodeficiency or to an autoimmune disease. So it's either defective, having low function, low immune function, or an abnormal, uncontrolled immune function, which is an autoimmune disorder. For example, myasthenia gravis. Myasthenia gravis is a disease that is characterized by severe muscle weakness due to an autoimmune destruction. So you needn't know the details of myasthenia gravis, but it's just related to dysfunction of the thymus. Not all cases, of course, of myasthenia gravis are due to thymus dysfunction, but some of them are. The appendix, as we said before, helps two things. It helps the immune response because of the presence of, because it is a secondary lymphatic organ, and it's also a safe house for storage of friendly flora. So, for example, so you can, like, if you want to generalize the functions of the appendix, then it aids in the immune responses because it is full of the wall, the mucosa is full of lymphocytes. But it's also, from grade 10 that if you take an antibiotic, for example, this would kill the friendly flora and disturb the ratio of friendly flora to pathogen. So in such a case, the appendix pumps those friendly flora that it had already stored in the colon in order to replenish your stores of friendly flora. Just like the bone marrow is, unexpectedly, the bone marrow, although it is a secondary organ, it's still considered a secondary organ, yet it plays a role in maturation of some of the B lymphocytes. But of course, the role is a secondary role because it's not responsible for the maturity of a large amount of B lymphocytes. What's interesting about the bone marrow, so we mentioned several things about the appendix. Number one is that it has an immune function because it contains lymphocytes and because it helps in maturity of the B lymphocytes. It also helps establishing balance of the gut flora whenever you have an imbalance in the gut flora, but it's also important for local immunity, not only for systemic immunity. It is important for local immunity because some immune cells in the mucosa of the appendix secrete antibody, which is an antibody molecule which is called immunoglobulin A, and immunoglobulin A is called surface antibody. Surface antibody. So it is important for all the surfaces of the body. So you have immunoglobulin A in your oral mucosa, you have immunoglobulin A in the inner mucosa of the trachea and the bronchi. You have immunoglobulin A in the inner mucosa of the small intestine and the appendix. So immunoglobulin A is a surface antibody responsible for what we call mucosal immunity. So locally, if you have any pathogen on the inner mucosa of the appendix, then the presence of this immunoglobulin A will help attack that pathogen and kill it. So a third function for the appendix is mucosal immunity. Um, the lymphocytes in the appendix are just like in any other secondary organ, present in the form of lymphatic follicles, those spherical collections or aggregations of lymphocytes, and those lymphatic follicles are called Peyer's patches. So what are Peyer's patches? They are lymphatic follicles in the mucosa of the appendix, the small intestine, that are full of lymphocytes. So in the past, it was said that the appendix is smaller in size in human beings than in herbivores because it's not used, because humans do not digest cellulose, and therefore their appendix does not produce cellulase enzyme, because you know that rabbit's appendix produces cellulase enzyme to digest cellulose since their diet is full of cellulose. But now it has been proved that the appendix is not a rudimentary structure, is not an atrophic structure. It is a structure that is very important. The last thing I want to say is concerning immunoglobulin M, immunoglobulin A, and immunoglobulin G. All these are antibody molecules, meaning that they are peptide or protein molecules that can attack antigens, that can attack foreign antigens or non-self antigens. But what's the difference between them? We have immunoglobulin M is secreted upon first exposure to an infectious agent or a foreign particle. Immunoglobulin G is produced later on or upon secondary exposure, and immunoglobulin A is only local mucosal, is only responsible for local mucosal immunity. So the first exposure involves immunoglobulin M, which is a rather big molecule. The second exposure develops more that develops later on upon secondary exposure will give rise to immunoglobulin G and not to immunoglobulin M. Thank you, guys, and I wish you the best of luck tomorrow, inshallah, in your chapter test.